Hi, my name's David Julius. I'm in the physiology department at UC San Francisco. And I'd like to take a few minutes to tell you about how we have used spices to understand mechanisms of temperature and pain sensation. And I thought I'd begin just by telling you a little bit about things that have interested me and what sort of inspired me to work in this area. So for many years, I've had an interest in natural products and how these can be used to explore physiology and neuropharmacology, and to wonder how people have discovered the use of plants through folk medicine and other ritualistic behaviors, how the active ingredients were identified, and how they've been used in biology to explore signaling pathways. And I just put this slide together to show you some of my heroes in this area and how they've used natural products from things like opium poppies, psilocybin, or marijuana to understand key signaling pathways in the brain, like opiate receptors, serotonin receptors, and cannabinoid receptors, and to illustrate the power of natural products for identifying endogenous signaling components in our nervous system. The other thing that I've been interested in for many years, like many people in neuroscience, is to understand sensory systems. And I think for many of us, including myself, this fascination really comes from the idea that sensory systems really determine how we perceive our world. The kinds of frequencies of light we can see, the frequency of sounds we can hear, the things that we can taste are really determined entirely by the sorts of receptors that we have in our sensory systems and how that information is integrated in our neural circuits. And what's also interesting is that different animals see the world in different ways because they have different receptors. A rattlesnake, for example, can see a rodent in a moonless night because it can detect its body heat. And so I think this raises fascinating questions about evolution and about how biophysics has tuned sensory systems to enable us to each live in the environment that we need to live in. Some years ago, we decided to merge these interests. I decided to merge these interests and to study an aspect of sensory systems that at the time was less well understood than, say, chemosensory systems like olfaction, taste, and certainly vision. And that is to understand somatosensation, or our sense of touch, and in particular, to understand this in relationship to how we experience pain. And the aspect of pain sensation that we focused on is nociception, which is the initial act of detecting a noxious stimulus. So nociception is to pain, for example, what phototransduction is to vision. And we've been interested in a subclass of sensory nerve fibers called CNA delta fibers, the so-called nociceptors, that are the subset of somatosensory neurons that carry out the bulk of detection of noxious stimuli and contribute to pain sensation. And our goal was really to identify molecules and mechanisms that enable these nerve fibers to detect things like environmental stimuli, changes in ambient temperature or pressure, or noxious chemicals, or even endogenous things that are produced during inflammation, endogenous chemicals like prostaglandins and other things that generate or contribute to pain sensation. And the place where this all comes together for me is really that natural products have played such a key role in trying to understand how these nerve fibers work. And probably the best example is given by pungent agents from capsicum peppers or hot chili peppers. And back in the 70s, Jansko and colleagues in Hungary showed that the pungent ingredient in hot chili peppers, namely capsaicin, elicits a sensation of burning pain by serving as a specific chemical activator of these nerve fibers that are involved in pain sensation, namely the nociceptors. And some years later, Bevan, Rang, and others in London carried out some biophysical studies to suggest that what capsaicin does when it interacts with nerve fibers in your mouth or anywhere else in your somatotopic surface is to depolarize the nerve fiber and allow sodium and calcium ions to flow in. And the main questions that were still unresolved by this was, does capsaicin interact with a specific site, a so-called vanilloid site, because capsaicin is a vanilloid compound, like other pungent agents in peppers? Is there a specific receptor for capsaicin? If so, what does it look like? And what might its normal physiologic function be in the body? What role does it serve in our endogenous ability to sense pain? And we, like many other people in the field, realized that identifying or addressing these questions could lead to some key insights into pain sensation, because sensitivity to capsaicin was a major functional hallmark or characteristic of these specific nerve fibers that are involved in pain sensation. In many ways, it was to the nociceptor what the T cell receptor was to lymphocytes. And so we set out to do this. But the problem was that nobody really knew what this receptor might look like, even if it existed. And so we decided to take sort of a structure-blind, identity-blind approach to doing this by just exploiting some functional attributes. And the idea was that if a capsaicin receptor does exist, you could make a cDNA library from sensory nerve ganglia and introduce those clones into a non-neural cell that's normally capsaicin insensitive, such as a fibroblast. And then if you expose these to capsaicin, and that allowed calcium to flow into the cell or to somehow be released from intracellular stores, you could detect this using calcium-sensitive dyes, such as those developed by Roger Chet and others like Indo or Fura. This has inherent risks. The advantage is that you make no predispositions about what this receptor, mythical receptor, might look like, but the risk is that if the receptor is, say, encoded by multiple gene products, the ability to transfer a single gene to a fibroblast and recapitulate this activity would be very difficult, if not impossible. But you can't do science without luck and some risk, and that's kind of what we're all in this business for in some ways, to do something new and exciting. And so we tried this, and lo and behold, we ended up by this method identifying a single cDNA that encoded for a capsaicin receptor, which we call the vanilloid receptor. And this is work done by a wonderful postdoc in my lab at the time, Mike Caterina, who now has his own group at Johns Hopkins University. And we could show that this clone gene encoded a functional receptor, because when you introduce it into these cells and use patch clamp electrophysiology to look at what's happening, we could show that when you put on capsaicin to these cells, in fact, you induced inward current flow of sodium and calcium ions into the cell. And so this told us that the TRPV1 that we had identified, this gene, is in fact a vanilloid activated channel. But Makoto Tamanaga and Mike Caterina and others in my lab then addressed really the most interesting question, which is what else does this receptor respond to, and what might its normal physiological role be in our ability to sense noxious stimuli. And we tried to activate this receptor with all kinds of things that we knew generated pain psychophysically, or activated sensory neurons in culture or in nerve fibers. And we went through a whole gamut of things, neuropeptides, other neurotransmitters. And it was only after some time that we realized, gee, these nerve fibers detect changes in our environment as well as changes internally that are related to pain sensation. And we should try some of these things. And the one that we tried that worked was heat. And in retrospect, this seems obvious because you describe a chili pepper often as a hot chili pepper. And this is a very nice parsimonious explanation of that. But this wasn't the first thing that we thought about. But shortly after failing to look at it, to find results with a number of other stimuli, it occurred to us that this might be the case. And it's really a beautiful result in that you see that the channel, when expressed in this case in a frog oocyte, is silent until you heat the solution, the external solution up, and then the channel opens and you see this large ionic current go through the cell with a threshold that is very consistent with the sensitivity of heat-sensitive nociceptors to heat, and in fact is very close to the threshold that most of us would describe as the demarcation point between determining that something is innocuously warm and noxiously hot. So this has led us to propose that TRPV1 is a little molecular thermometer that sits in the membrane and at least contributes, maybe doesn't underlie all of, but contributes to our ability to detect ambient temperature in the warm to hot range. Now, we all know that we can sense temperatures over a wide range, including cold, not just hot, and how might this work? And we thought if we could figure this out, we could maybe get some larger view of how temperature sensation works, and ask if our ability to sense temperatures at different degrees is due to sort of a similar and unifying molecular concept, or represents different molecular mechanisms. And again, we wanted to take an approach that was not biased. We didn't want to look for channels and genes that were similar to TRPV1, but instead step back and ask, how is cold sensed from just a functional point of view? And again, the great way here, we turn to natural products, because we all know that, that cooling agents that are derived from mint leaves or eucalyptol produce a sensation of cooling. And Hensel and Zoderman in the 50s in Sweden showed that this was the case in neurophysiologic recordings of sensory nerve fibers. So we played the same game that we played with the capsaicin receptor to ask if we could identify a menthol receptor. And the question is there a menthol receptor? If so, does it respond to cold? And is it related to TRPV1, or is this a totally independent molecular mechanism? And so we did, meaning David McKemey, a postdoc in my lab at the time, and Werner Neuheiser, ended up identifying a clone and a molecule called TRPM8 that, when expressed in fibroblasts renders them sensitive to menthol and other cooling agents. And as you can tell from the name, TRPM8, it belongs to the same family of so-called TRP ion channels as does TRPV1. It's a molecular cousin. And the question is, does it respond to cold? And in fact, again, when you express this in a frog oocyte or some other cells, you can see that the channel is quiet at warm temperatures, but then opens when you cool the cell below about 26 degrees. And so this led to this idea that there is some unifying concept of how we sense changes in ambient temperature, and that TRP channels serve as, or at least contribute to, our ability to detect changes in ambient temperature over a pretty wide range, and they act as little molecular thermometers in the membrane that initiate the depolarization of a nerve fiber in response to changes in temperature. And the best way really to test this, of course, is in a living animal in genetics, using genetics. And I think the clearest demonstration of this really comes from knockouts of TRPM8, which show a very beautiful phenotype in terms of deficits in thermosensitivity. So we made a mouse that lacks this channel. This is work done by these wonderful people in my lab, Jan Siemens, Diana Batista, and Sven Jurt, who now all have their own labs in Heidelberg and UC Berkeley and Duke University, respectively. And they set up a very nice, simple paradigm to look and see if deletion of this gene has a phenotype associated with temperature sensitivity. So here you do this very simple test where you take, in this case, a wild-type mouse, and you ask if it can discriminate between a warm platform and one that's somewhat cooler, namely 20 and 30 degrees. And you can see without any training, this animal avoids the cool side and likes to stay on the nice, warm, cozy side, as is true for most of us. And he'll stay on this side all day long. Now, if instead you play this game with a mouse in which the gene for the menthol receptor is knocked out, TRPM8 deficient mouse, you'll see that the animal cannot discriminate between these two platforms and will spend equal time on either one. And in fact, it won't be able to discriminate between platforms even when the cool side goes down to very low temperatures, to about 10 degrees. Below that, the animal can detect changes, can detect a preference for the warm side, which may be due to other processes that contribute to cold sensation. But in any case, I think this validates the idea that TRP channels, and in particular TRPM8, play a really important role as a molecular detector of ambient temperature. So through these studies, we've been able to identify key elements that enable cells, that really enable cells to specify their sensitivity to environmental stimuli. And as in other sensory systems, one of the great things about doing this, identifying genes and molecules that specify the sensitivity of cells and nerve fibers to stimuli, is that you can use them as molecular probes to try and understand the cellular and circuit logic as to how this information is conveyed to the nervous system and ultimately to your brain, so that you can discriminate different percepts, in this case heat from cold. And what these markers, antibodies to these channels, now tell us is that the cold and hot receptors are really segregated. They're expressed by distinct subpopulations of sensory nerve fibers, which tells us, at least at the outset, that the information as it comes into the spinal cord, at least at the beginning, is segregated. And that information about cold and information about heat is conveyed by two so-called labeled lines, separate lines of sensory nerve fibers. And this at least begins to give us some idea as to how behavioral specificity is dictated by by circuitry within at least the peripheral aspect of the nervous system. So we've used natural products to identify some important players in pain sensation, some of which I haven't had time to tell you about today. And all of this work has really come about through just sort of curiosity-driven research, which, by the way, was funded by you, the taxpayers, the NIH. And we've just been curious about how this system works and allows us to appreciate the world around us. But as so often the case, curiosity-driven research can have very practical and important therapeutic aspects, and all of these channels are now novel targets for the development, hopefully, of new types of analgesic drugs that we can use to treat a variety of peripheral pain syndromes. And that's work that's in progress by a number of pharmaceutical and academic labs. But hopefully, our goal to understand how the system works at a curiosity-based level will also have some practical outcome that will benefit patients who suffer from chronic pain syndromes. And finally, I'd like to say that my lab is small. There are only maybe about, or relatively small, anywhere between 7 and 9 people at any given time. And so the way that we cover the waterfront, I'm a big believer in trying to hit a problem from multiple different angles. And in our lab, we try and do this starting from molecular biology through biophysics, through animal behavior and anatomy and physiology and ultimately to structural biology. And the way that we do this is to work with other fantastic scientists through close collaborations, some at my own institution, some other places in the world. And this has really allowed us to expand our scientific horizons, which is something that I think every scientist should try and do, and to work outside your comfort zone and really stretch yourself. And of course, all these people are now great friends, and this is a wonderful way to meet people and have lifelong friends, and also enlarge the sphere of people who folks in your own lab can interact with. So I'm a big believer in collaborative science, and we've had a fantastic time working on these projects within my lab and with many friends around the world. Thank you very much.